Me toca ser presentador, Santiago no podía venir y Martín está de viaje. Pero es un gusto presentar a Boris-Marko Kukovec. Él es de la Universidad de Split en Croacia. Él ha venido ya en una visita en el 2023 por 3 meses. Esta es su segunda visita, también por 3 meses en 2025. O sea que le gusta mucho nuestro lugar y Buenos Aires. Y bien, ahí nos va a hablar sobre, como dice en la transparencia, polímeros que él sintetiza con ligandos nicotinato y diferentes metales, principalmente níquel, cobalto y paladio 2. Nos va a hablar de eso y bueno, también quería decirles que si al final a alguien le interesa por alguna posible colaboración o por preguntar alguna cosa en particular o lo que sea, después del seminario se puede acercar y hablamos y vemos, se quedan unos días más, así que puede tener alguna reunión con alguien que pueda estar interesado. Así que, thank you, Boris.
Thank you, Fabio,
I'll let you talk.
for your kind introduction. Ladies and gentlemen, good afternoon. Fabio, I'm also grateful for this opportunity to present my work. So I'm happy to be here again after my first visit in Buenos Aires. But Fabio already told you all that. So yeah, he's totally correct. So I'll start with my lecture. So the title, you can see the title there. But first, some general points about the place I'm coming from. So Europe, you are familiar with Europe, so the countries that you are probably the most familiar with, Spain, France, Germany is here, then Italy, and where is Croatia on this map? So it's here, and you can see it better here, so Croatia, yeah. It has a strange shape due to historical reasons, but I'm not going to talk about the historical reasons. So the country is, by Argentinian standards, quite a small one, by population and by area as well. So the capital is Zagreb, it's somewhere here, and I currently live in Split, which is here at the sea coast. So here is the Adriatic Sea, which separates Croatia from Italy. So the Adriatic Sea is here, so Italy, Croatia. And to better realize how small the country is, for instance, from Zagreb you can travel to Hungary, and in 300 kilometers you reach the Hungarian capital. If you go more to the west, in 300 kilometers you end up in Austria and in the capital of Vienna. If you want to travel to Italy, of course you can, it's also very near. For instance, from Split there is a boat which goes to Italy in, I think during the night, and you are there by boat, which is not very fast. So, and for instance, if you want to travel somewhere in Europe from Zagreb either to France or in Germany, you are there in a 2-hour flight, even less than 2 hours. So compare here, if you fly in 2 hours, you are still in Argentina by far, right? So comparing to Argentina, the whole country is very, very small, including the whole Europe. But okay, so that's about the location. Split, the city I currently live in, it's the second biggest city in Croatia by population. So it's located on a peninsula. So this is the Split Peninsula, and it's so surrounded by sea from three sides, here, here, and here. So what I want to say. So yeah, the city center is situated somewhere here, and then the city is spread across the whole peninsula. And here you have some forest and the mountain by the hill. So what is interesting here? So the city center, the Diocletian Palace, shown here, it's around 2,000 years old. So 2,000. So it was built in in the 6th century after Christ. So this is only one plaza in the Diocletian Palace. This is the Split town cathedral at the moment, and it was the Roman temple in the past. So here you can see some views of the Split. So yeah, I'm not going to talk about these figures these pictures in particular, but just to have some visual impression how it looks like. And this is the old part, and here this building is the rather new one. It was built 10 years ago, so in 2015. So it comprises 3 faculties. One of the faculties is the one that I'm working at, that's the Faculty of Chemistry and Technology. So we have, I think, 2 floors in this building, you can see it here. So it's also the part of the campus, so there are other buildings that belong to the University of Split. And in this picture you can see the regular student laboratory, for some laboratory course, so how it looks like. So since the building is relatively new, all the equipment there is also purchased in the last 5 or 10 years top. So, okay, that was the short introduction, non-scientific one. And now here is the plan of my today's lecture. So, as an introduction, I'm going to say something about coordination polymers in general, about the definition, about the dimensionality of the polymers, 1D, 2D, 3D, what are metal organic frameworks, and about the properties and applications of these compounds. So why are we interested in these type of compounds? And afterwards, something about heterometallic coordination polymers, how are they prepared, and what is the application, and finally about the ligands used, the ligands I used for the preparation of these coordination polymers. These are substituted nicotinate ligands and some selected auxiliary bridging ligands. And then finally, I'm going to talk about 3 chapters of my work. The first one is will be about one-dimensional cobalt polymers with halonicotinate ligands, then cobalt or nickel polymers with hydroxynicotinate and aminonicotinate ligands, and finally two-dimensional heterometallic sodium-palladium coordination polymers with the same type of ligands, in this case nicotinate ligands. So here's first something for the introduction. The introduction is going to be as short as possible, just to have some more insight about this topic. So coordination polymers, what are they in general? So these are infinite systems constructed by metal ions and organic ligands as the main building units. What is important to say here, the metal ions and ligands need to be connected exclusively by covalent bonds in order to be a coordination polymer. So covalent bonds between the ligands and metal ions are crucial, and dimensionality of the polymers can be either 1D, 2D, 3D. So that refers to the polymer spreading in these dimensions. And finally, you probably heard for metal organic frameworks, how are they connected with the polymers. So polymers, metal organic frameworks need to be 3D coordination polymers, but they need to have additional solvent or gas accessible voids in the structures. In other words, these frameworks need to be porous, and in order to be called metal organic frameworks. But these are just not very strict definitions, so it's rather loose. And finally, why are these compounds interesting? So the applications varies from catalysis, gas and energy storage, then gas separation, magnetism, luminescence, molecular sensing, biomedical imaging, and many more. But I'm not going to talk about that in more details. So what about the one-dimensional coordination polymers? These are usually chains of various forms, and you can see these various forms in this slide. So if you look at these, so these spheres represent metals, and the sticks in between the spheres represent the ligands. So if you look at the arrangement of the spheres and the ligands, it can be like this. So it's just a linear one. Then if it goes like like this, then it is a zigzag, two types of zigzag, then it can be helical as well. It can be a ladder or a double chain if you look at this, it can be a ribbon. Even the metal clusters can be connected by ligands into a 1D polymer. What about the 2D coordination polymers? In this case, the networks formed have various topologies. So some of the most frequent topologies are a square grid, for instance, then a rhombic grid, rectangular grid, honeycomb grid, brick wall, herringbone, etc. So in the case of the networks, we can have different topologies. And finally, a 3D coordination polymer, as I mentioned in this figure, a metal is represented by a red sphere, the ligands are represented by these black, if the polymer spreads in the 3 dimensions, like you can see here, 1, 2, and the second, then we get the 3D polymers, and of course, if there is voids in there which can be filled with different molecules, then we are talking about the metal-organic frameworks. So I also mentioned the heterometallic coordination polymers. What are they? So in case we have more than one type of metal ions in the structure which are connected together, then we are talking about heterometallic. And why put more than one metal in the same structure, in the same compound? Then in that case we have the synergistic and cooperative effect of these metals on the functionality of the resulting compound. These heterometallic coordination polymers have various applications as a heterogeneous catalyst and in photochemical and electrochemical reactions, and also as a precursors for obtaining mixed metal oxide by thermal decomposition. How do we prepare these heterometallic coordination polymers? So it's rather simple. We usually use the self-assembly method in most cases under hydrothermal conditions or at atmospheric pressure. So we just combine different metals and the ligand in a solution, and we have either solid thermal condition or a crystallization at atmospheric pressure, and we get the heterometallic coordination polymer. Here you have this one looks to be 2D, and we have two different metal ions here, represented by different colors in the figure. Okay, so what are the ligands that we use for preparation of coordination polymers? So halonicotinate ligands, so it's the nicotinic acid with the addition of halogen atom at various positions. So we had at position 2 either chloride, bromide, then bromide at position 5, and the position 6 of 3 halogens, fluoride, bromine, and chloride. We also use different substituents like methyl, hydroxy, and amino substituent. So we have at position 2 methyl group, at the position 6 both hydroxy and amino group. I also mentioned the auxiliary ligands, the bridging ligands, which were used as spaces in our case. So from the most simple one, 4,4-bipyridine, to more complex one, 1,2-bis 4-pyridyl ethane, or the similar compound, but this one is not saturated. So this is 1,2-bis 4-pyridyl ethane. So 1D polymers I'm going to talk about were prepared by using cobalt ions and the nicotinate ligands from the previous slides and the bridging ligands shown here. And for the 2D polymers we use palladium ions and sodium ions, but just the nicotinate ligands, that was more than enough to get the 2D polymers. Okay, so now I'm continuing with the first chapter. So the first chapter is one-dimensional cobalt-2 coordination polymers with halonicotinate ligands. So the synthetic route for these two compounds is as shown here. So we used cobalt acetate, then the auxiliary 4,4-bipyridine, and these two ligands, nicotinic acid and 5-bromonicotinic acid. So the halogen atom is placed here in the position 2 and here in the position 5. Reaction conditions were quite similar, so the ratios of the reactants are given here. I'm not going to read it. We used both the mixture of ethanol and water as a solvent, and the crystallization was done at atmospheric pressure, so nothing too experimentally demanding. So we get two different coordination polymers. In the case of 2-chloronicotinic acid, we got this one, and the second one in the reaction with 5-bromonicotinic acid. So now we'll have a look at those compounds. So in the case of this one, so the asymmetric unit is shown here. So we have the octahedrally coordinated cobalt ion. The chloronicotinate ion is only an O-monoethane ligand, so you can see it here. We also have two coordinated water molecules in transposition, so these two molecules. And for bipyridine acts as a bridging ligand. You can see it here, so two of them coordinated to cobalt, and there are also some lattice water molecules, but that's not really important. The second one obtained by using 4-bromonicotinic acid was a bit more complex one, because here we have the complex cation, we also have the complex anion, and we have some lattice for bipyridine and water molecules. So the cation is a polymeric one. It's shown here. So cobalt ions are bridged by 4,4-bipyridine ligands, and there is also 4 water molecules coordinated to a cobalt ion. And as an ion, so this is a cation. As an anion, we have a discrete cobalt complex composed of cobalt ion, shown here, coordinated with 4 5-bromonicotinate ligands. So the first one, second one, third, and the fourth one. So 2 of them are N coordinated, and the other 2 are O coordinated. And as I said, we also have additional, you can see it here, co-crystallized or latticed 5,5-bipyridine ligands and water molecules. So if you want to compare these two polymer chains from the first and the second one, you see in the one with the 2-chloronicotinic acid, the 2-chloronicotinic acid is coordinated as an ion monodentately to the cobalt, and the cobalts are bridged by 4,4-bipyridine ligands molecules. In the second one, which I said the polymeric one is the cation, it's more simple to this one. So yeah, the cobalt ions are bridged by 4,4-bipyridine ligands, the same as up, but here we don't have coordinated 5-bromonicotinic ligands, so that's the main difference. So for these two compounds, we also did some DFT calculations in order to shed some more light on these different behavior of compounds obtained by 2-chloronicotinic acid and 5-bromonicotinic acid. So in general, I didn't put any numbers here because I take it you are not very interested in the numbers, but just to the general conclusions. So how substituent and the nicotinate ligands, in this case chloride or bromide. So the type of the is it chloride, is it bromide, it doesn't have much effect. So but the position at the nicotinate ligand, in particular position 2 or position 5, that position has the main influence on the cobalt coordination environments in the obtained polymers. So in general, the substituent position is responsible for, in these two cases, for different structural types of cobalt coordination polymers. After that, we did some more studies, but in this time we wanted to exclude the water as a solvent, so we used exclusively DMF. Okay, DMF maybe contained some water, but it was not distilled, not dried, I mean DMF. So we used again the cobalt salt, but instead of for bipyridine we used the bipyridine ligand that I mentioned on one of those previous slides. Yeah, and in this case we had 6 chloronicotinic acid and 6 bromonicotinic acid. We also got two, when you look at this, two different coordination polymers. So if you look at them, these are the asymmetric units. So in both cases, the cobalt is octahedrally coordinated. Halonicotinate ions, be it 6 chloronicotinic or 6 bromonicotinic acid, are coordinated as a chelate ligand, and that's this one and this one, and the second one is coordinated. This one is the bridging one because it goes. This one bridges another cobalt, which is not shown here. So we have both bridging and the chelate, and also the bipyridyl ligands. This one and this one also acts as a bridging ligand in another direction to get the 2D polymer. Sorry. Not the 2D, but the double polymer. You'll see that in the next slide. So yeah, since we didn't do this synthesis in water, there is no water molecules, of course, either coordinated or lattice molecules. So when you look these two compounds, you see that we got, yeah, so this can be called either a ladder or a double chain. So I call it here a double polymeric chain. So 2 cobalt ions are bridged with 2 bridging halonicotinate ions. So this is one bridging the other bridging. We also have 2 chelate ones only attached to 1 cobalt. So in this way we get the dimer, and these dimers are further connected by bipyridyl ligands. So in this case, each dimer is connected by a pair of bipyridyl ligands. So if you look these two polymers, on this figure, they look quite the same if you look at them. But what is the difference? In the case with the 6 chloronicotinic acid, we use the 1,2-bis 4-pyridyl ethylene, and in the case with 6 bromonicotinic, we use the 1,2-bis pyridyl ethane. So this one has a double bond, this one has only a single bond. Due to a double bond here, ligands, this one and this one, this one and this one, are not parallel to each other because there is no rotation about the double C bond in this case. But in this case, when we have only a single bond, there is a rotation, so that bond is here. So there is a rotation about these two bonds, and these these ligand and these ligand in that case are not parallel like at the other example. So now these were the selected polymers obtained with halonicotinate ligands. We also used some different, as I said previously, different substituents like hydroxy and aminonicotinate. So here we have the synthesis of both nickel and cobalt coordination polymer, but this time with 6 hydroxynicotinate ligand and the most simple 4,4 bipyridine. So the synthesis here was different from anything that I mentioned previously. So we again did it in the mixture of both ethanol and water, but we used hydrothermal synthesis, hydrothermal conditions here. So the reaction mixture was held at 130 degrees for 3 days. So we put 4,4-bipyridine, the hydroxynicotinate ligand, and either cobalt or nickel salt, and we got almost the same type of compounds here. The difference is here is the cobalt and here is the nickel, and they are in different colors just for you to follow it more easily. So if we look at the asymmetric unit, and we can, the symmetric unit is here. So we concluded that both nickel and cobalt polymers here are isostructural compounds. So what isostructural means as a crystallographic term? So it means that if we look at the asymmetric units, the positions of the analogous atoms are the same. So if we here I just put the cobalt one, the nickel. So if you overlap the cobalt and nickel one, you get almost the same structure. That means the isostructural. So the only difference here between the nickel and the cobalt one is the metal overlaps cobalt either nickel in this case. So regarding this coordination polymer, the cation is the same as I mentioned previously. So it is the cobalt coordinated with bipyridine ligands and 4 water molecules, and the anion is just hydroxynicotinate anion, which is not it's not bound. Just it's not bound to any metal. So it's not coordinated, and we also have some lattice water molecules here. And as you can see, here is the cation. It is the same one as it appeared in some of the previous examples. So it's an infinite one-dimensional polymeric chain. Cobalt ions are here. They are connected with 4,4-bipyridine ligands. You see the water molecules. The adjacent cobalt ions, this one and this one, are bridged with a single bipyridine ligand. So as I said, we have cobalt and nickel in the same synthetic conditions. Crystallography revealed they are isostructural, so we wanted to check if there are any difference in the end for these two types of compounds. So we did the cyclic voltammetry study on these two ones, but not the regular one. So we used the multi-volt carbon nanotube electrodes. So the compound, either nickel or the cobalt one, were dispersed in these nanotubes, and that served as the electrode. And we used the the cyano coordinated hexacyano ion 2 and 3 their mixture as electrode probe. And we observed, so as I said, these two, nickel and cobalt, they are isostructural. But we observed a different reactivity of both of them towards these redox reaction, because here you have, so this is either ion 2 or 3 in this redox probe. So if you look at it, this the black one is just the black one. This is just the multi-volt carbon nanotube electrode, and the red one is the cobalt, and the blue one is the nickel one. And you can see in the case of the cobalt one, there is no redox activity because the peak currents are even lower than the multi-volt carbon nanotubes without anything. And in the case of the nickel one, we got the additional peak, which was assigned to the oxidation of ion 2 in this case, and a reduction of the nickel in that compound. So inspired by these results, we used again in the previous example we had 6 hydroxynicotinic acid. Here we have 6 amino-nicotinic acid, not for bipyridine, but this bipyridine ligand, which is a bit more complex one. So this is, as I mentioned, 1,2-bis for bipyridyl ethane. So we reacted these two with the cobalt, again using hydrothermal synthesis for 72 hours at 130 degrees, and we get again a coordination polymer of some type. You can see it here. So again we have a 1D polymeric cation, but which is different from the one I described previously. You'll see afterwards. The cobalt coordination is again octahedral. We have 2 coordinated water molecules, so they are here in transposition, and this time 4 BPE bridging ligands. So this one, here and here, and so that's only the cation, and the anion is again just the amino-nicotinate ligand, which did not coordinate, and some nitrate ions and lattice water molecules are also present there. So if we look how this chain, this 1D chain looks like. So again, the cobalt ions are bridged with, so these two and these two are bridged with 2 4-bipyridyl ligands. You can see it here, and in this case we get, if you look at here, we have 22 membered microcyclic rings. So if you want to count, there are, in this ring there are 22 atoms, or you can just believe what I say, don't need to count. Again, we use the cyclic voltammetry to see if we can use this compound for some analytical purpose, and this time we were lucky because it, before I explain why we were lucky, so we used, in this case we used modified glassy carbon electrode, again mixed with the cobalt polymer which I explained, and we reacted, we did in this time with dopamine. Why dopamine? Because dopamine is essential, so the structure of dopamine is shown here. So it's rather, it's not a complex molecule, that's what I wanted to say. So it's essential neurotransmitter, it's crucial in medical treatment and clinical analysis, and regardless of many analytical methods to determine this dopamine in biological samples, there is a demand for more and more precise methods for analytical quantitative determination of dopamine. So that's why dopamine. So we used different concentrations of dopamine, and this electrode which was prepared as I explained, and you can see the results here. So what are the results? So if so you see the concentration of dopamine is increased here. So if you look at it here, it's a smaller concentration, the higher concentration, you see that the peak currents increase with the increasing the concentration of dopamine in the solution. So, and that was the linear increase. So, judging by these results, it seems that our compound described here can be used as an electrochemical sensor for dopamine. However, cyclic voltammetry was not good enough to determine the exact analytical method for the determination of dopamine. So that's why we used square wave voltammetry, which was more efficient for this purpose. So in this case, we managed to electroanalytically determine dopamine. And so how it worked. So this is the square wave voltammogram. In this case, you also have various concentration regions, and So I wanted to say first we have two peaks in the voltammogram. The same two peaks correspond to the same process, the oxidation of dopamine, but the first peak corresponds to the oxidation of the dopamine which is attached to the surface of the electrode, and the second one is the free dopamine in the solution. So how did we conclude that? So if we increase the concentration of dopamine from 0 to 10 minus 5 mole per liters, so we have the peak increase of the peak current increases for the first peak, but after, so that's the left peak. So but the left peak. And if we increase the dopamine concentration beyond 10 minus 5, from 10 minus 5 to 10 minus 3 mole per liters, we see that the peak current of the second peak increases. So, and this one doesn't increase more. So, as we explain it, so from until 10 minus 5, there is a peak current increase here because the surface of the electrode is still available, but at 10 minus 5 it is saturated, so that it doesn't increase more, and the dopamine starts to oxidize, but not that one at the surface of the electrode, but the one in the solution. So if you look at the linear function of the peak current and dopamine concentration, it's shown here. This one is for the lower concentration of this peak, and this one is for the higher. So both peaks can be used, but depending on for the analytical determination, but depending on the concentration range. Okay, so the final chapter. So I was talking about 1D polymers with various nicotinate substituents, but now I'll show some examples of 2D polymers, which I mentioned in my introduction. So we used previously mentioned 2-chloro-nicotinic acid and 2-bromonicotinic acid. This time the palladium salt, sodium tetrachloropalladium palladate 2, yes, and we also add the additional, what is this, hydrocarbonate as a base, and it was done in the water, and we got two interesting nets. Yeah. So why are these 2D polymers? Before that, we also used this is still not published, but currently in preparation, but I included here, never mind. So we also used 2-methyl-nicotinic acid and 6-fluoronicotinic acid, and very similar experimental conditions. The only difference is here, if we use the 6-fluoronicotinic acid, we use the mixture of ethanol and water, and a little, somewhat different ratios of the reactants, but it doesn't matter at this point. So here, in this figure you can see the asymmetric units. So first one is 2-chloronicotinic acid polymer, and the second one is 2-bromonicotinic acid. Here is the overlap. You can see they are quite similar. The difference is there is the bromide coordinated to palladium in the bromonicotinic one, and the chloride in the fluoronicotinic one. Sodium ions are 5 coordinated, octahedrally coordinated, but with 5 water molecules and the carboxylate oxygen from nicotinate, and the palladium is coordinated by nitrogen atom from the aromatic ring. Yeah, so both 2-fluoronicotinic and 2-bromonicotinic are bridging ligands here. They bridge sodium and palladium. So as I mentioned, these are just the asymmetric units. In the case of the methyl, this one is the 2-methyl nicotinate polymer and 6-fluoronicotinic polymer, nicotinic ligand, yeah. They are more or less very similar to the previous one. I'm not going to mention it, okay, but in this case you can see they don't overlap that nicely because all this part, this is the sodium coordinated with water again. These two do not overlap in methyl and fluoronicotinate compounds. So how do these 2D networks look like? You can see it here. This is, as it says here, 2-fluoronicotinic polymer, and this one is 2-bromonicotinic polymer. So here we have two main building units. First building unit is the water bridge sodium ions. So these yellow ones are sodium ions bridged by water molecules in both cases. So let's say that's the first building block, and the second building block is the palladium coordinated with 2 nicotinate ligands. So the palladium coordinated with, you can see it here, with 2 nicotinate ligands. So this is the second building moiety. So how do we get the 2D polymers in the end? So these chains built of the sodium ions, they are bridged by these palladium nicotinate moieties. So first chain goes here, and these chains bridged by these PD moieties in order to get the 2D network. So in the case of the 2-methylnicotinic and 6-fluoronicotinic acid, we get the same structural pattern. So I'm not going to repeat it because I just explained it, but what's the difference here? So if you look at this microcyclic ring, so this one between this moiety, this part of the chain, this part of the chain, here and here, in both cases 23-membered microcyclic ring, but in the first case this one is more like a rectangular shape, and this one, it looks like more like a parallelogram. So that's the first and striking difference that you can see when you look at it. So for now we did some DFT calculations, but only at this point on chloro and bromonicotinate polymers, and with the help of these DFT calculations we concluded that the Howard substituent type, be it chloride or bromide, they had no influence on the formation of either trans or cis isomers around palladium. So in all cases we got the trans isomers, and the theoretical trans isomers are 10 to 15 kilojoules per mole more stable than cis isomers. So this DFT result was in accordance with the structural result. And we also calculated energy stabilization for the polymers and the effect of Howard substituents, and in general we concluded that the different DFT calculated energies can be ascribed to different Howard substituents and the presence or absence of lattice water molecules in the structure. And for the methyl and fluoronicotinate, the DFT calculations are still ongoing. We also did some biological studies, in particular antibacterial and anti-proliferative activity of these compounds, but we weren't much lucky because there is no effect on Escherichia coli or S. aureus bacteria by these compounds, and there is also no anti-tumor activity in case of bladder or lung cancer cell lines. So we didn't go from there in this point due to these results. So here's a list of my collaborators, students, technicians. Yeah, some names you may recognize. Here the institutions involved, University of Split, University of Zagreb, University of Cape Town, Buenos Aires, the funding, and thank you for your attention.
Bueno, ¿alguna pregunta?
I have one. Thank you for your
Thank
talk.
you for listening.
Great synthesis work. And regarding the dopamine sensing, do you know which is the mechanism by?
We described it in a paper. I didn't put it here because it was too long, but I'm not. I need to look at the paper. But it has something with dissociation. There is some. It is the dissociation. The absorption of the dopamine on the surface is important in the mechanism. So we discussed the mechanism, but I'm not able to.
Okay, but
say,
it is in the paper.
It is in the paper which I cited. Yeah.
Okay, I wonder.
Yeah.
What was the proper.
Yeah. We describe it there.
I have one. When you show the cyclic voltammetries of the nickel versus cobalt, why do you think that nickel was being reduced by cobalt wasn't? Because the redox potentials are similar, right?
Yeah, but if you almost there. Yeah. So.
But I am just.
Why is that
why
so?
do you think it happens? Okay.
Well, because we didn't expect this. So we expected to be similar as nickel,
right.
but it didn't. It
Because
wasn't.
the structure is similar.
Yeah, exactly. Yeah.
And one just kind of general question. Any of these polymers that you have shown, have you thought if they could be used as catalysts for
they probably
for
could.
whatever chemical?
Yeah, they probably could, but we still didn't get that. So.
And catalyst for what?
I don't know, but no. I think I'm not sure for what, but probably they are because everything can be used. But whether if they will be efficient or not, that remains to be seen,
Sure.
right? But but since they are redox active as expected, I think they could be used.
¿Alguna otra pregunta? Agradecemos a Boris.